I'll open by saying that I actually come into this with a bit of my own bias. I'm Mike Watson. I'm Executive Director of the American College of Medical Genetics, but I spent most of my life as a laboratory director of genetic testing at Washington University in St. Louis. So I've lived through all of the attempts of FDA to get a handle on, not just FDA, all the regulatory agencies actually, to get a handle on oversight of genetic and genomic testing. And we're in a new phase of it now. And my bias is that I think we have LDTs for a very particular reason. When we were doing targeted tests, there was no incentive to industry to manufacture those tests because there was no way to get a return on investment with the clinical trial cost it took to bring a really rare disease test online. And I think that led to just the vast proliferation of these tests. So now we're at a stage where we don't just have to think about LDT regulation of clinical labs, but the much broader regulation of testing. If you look at the drug side of FDA, the Orphan Drug Act has done a very nice job of incentivizing industry to act in this space. We haven't had that on the diagnostic side. The humanitarian device exemption doesn't really apply very well to getting these tests, and the evidence is, based on the tests that we don't have, that it hasn't worked well. So there's a lot of aspects of regulation and oversight that I think we're going to have to address beyond just the LDT guidance about how to regulate the laboratories that deliver them. So on that, I will expand to one of the areas that I think is of great importance and is one we can talk about, and that's really data sharing. I think we're at a time now where there's going to be more genomes coming out of pharma than are coming out of the clinical laboratories. The numbers I've seen lately are pretty substantial. And the questions really seem to be whether or not the clinical data, the associations of a variant with a particular phenotype are pre-competitive or competitive. Are they something that really should be controlled, or is the need to have that information in the public domain to improve healthcare drive it into a more pre-competitive space? So would you like to address the area of data sharing and what's in this pre-competitive space versus is valuable in a competitive space without compromising healthcare?
So I'm not sure you can still call us a panel. We're two people now. Sitting up here. So I can say that, right, from our point of view, we have to be very careful about what we call pre-competitive versus competitive space, although we are involved in several consortiums to share information in that pre-competitive space. One of which is a pharmacogenomics consortium. We have some other consortiums where actual data is being shared. But I think it all depends, to some extent at least, depends on the quality of the data that you're looking at. If this is for investigational use only versus something that's actually generated at a clinical use level as a LDT, if I dare use that word. Or is this something that we're really looking at in an exploratory endpoint? So I think that's where we start to really think about the differences there. I don't see in the short term a model where we would share millions of exomes or genomes, for example. Maybe with the AstraZeneca model that changes a little bit. But it's hard for me to envision that right now in the current operating environment.
So I'm a clinical and a molecular geneticist, and I'll say that the first big change for us in terms of clinical diagnostics was when we were able to start seeing beyond just 1,000 genomes and really starting to see tens of thousands of genomes and knowing what normal and different variants were. So I do think in terms of being able to push that forward, at least on the diagnostics, being able to have more reference data to know what genes can tolerate variation, where in genes we can tolerate variation, and what are just normal ethnic differences is absolutely critical to a lot of the quote-unquote variants and genes of uncertain significance that many of us are sitting on these days. So I actually practice in New York City, up in Washington Heights, and for us, our local community is particularly disadvantaged. So we have a very multi-ethnic community that has a lot of African Americans, Hispanics, individuals from the Middle East, and I will say that we end up with, a bigger problem in terms of uncertainty with how to interpret their genomes. And so with the data sharing, I'm just going to advocate that we also be very expansive in terms of having many of our underrepresented communities in the United States represented. My fantasy is that what we're starting to do together with, for instance, things like the Precision Medicine Initiative and other things that are sponsored by NHGRI and NIH for data sharing will help us to be able to get through at least some of this. But as Mike was saying, a lot of this is going to be in other people's hands. And I think in general, the community of patients, at least the ones I represent, are very much in favor of sharing, just also making sure that we can protect patient privacy. So making sure that we do it in a responsible way, I think it serves everyone, and everyone is generally supportive, is what I would say, as long as we can protect the privacy.
So it may be that there's going to be a place in this continuum of what we know about a variant where data sharing is going to be of value to everybody. The more commonly understood variants, I don't think we need all that much help in clinical practice. But I'm one of the co-PIs in the ClinGen resource project, and what we're seeing across the genome and across genes is an enormously long tail of variants that are seen once or twice in people in the entire population that are much more difficult to get a firm handle on their pathogenicity. So it may be a place, somewhere in that continuum where we can all agree that we need to share about these singleton variants to better understand them, but that there may be other places of modifiers and things where that may be a little less critical, but we'll see over time. Now, we don't want to dominate this whole conversation, and I'd like to see if there's questions from the audience in the area of collaboration, regulation of LDTs. Otherwise, we will drive forward with some of our own questions. Wendy, yes.
Okay, so Wendy Rubenstein, NIH, and I work on GTR, ClinVar, MedGen. So one of the comments you just made was about the long tail of rare variants. And we've seen collaborations start with hospital systems and organizations that do sequencing and control, organized data. Is it not the case, though, that we're going to need to have those collaborations embrace the entire world, if you will, because there are so many rare variants, and seeing one, maybe even seeing two, isn't all that helpful. We're going to have to find all of them. So how do we wrap our mind around that?
I can tell you it's an enormous problem. We've been organizing some international projects. The Global Globe in 2020 initiative that's international. There's over 100 countries participating. And frankly, it boils down to almost a capacity problem in many countries of even bringing genomics online and bringing up their scientists and their healthcare systems to actually integrate it. So that's a significant rate-limiting step, but I agree with you that it's not just the variants, but it's the genomic backgrounds on which they operate that are going to be playing together and addressing these problems.
I mean, I'll just say, and you probably know more about this than I do with being in a large international, multinational company, but at least I've seen from many, both international research consortia and even more with pharma, in terms of being able to have different data privacy laws in different parts of the world. And so even though there's the will in terms of wanting to share data, from a regulatory point of view, they can't actually exchange data. So what I've been watching is that in individual countries, there will be a consortia where everyone will agree to put their data through a certain pipeline. Everyone will put it through a pipeline and then share the output of that pipeline, but not be able to put everything all in terms of being able to run the pipeline across everything. And I don't know if others have other experience, but that's at least what I've seen.
Yes, I will mention a bit about the problems we have running clinical trials in a global setting, right? We aim to have an informed consent document that would enable us to do this type of genetic research from all of our clinical trial participants. But there are certainly regulations in place in multiple countries around the world that prohibit that or restrict the access to that data / those materials. And I think we all are aware of which countries those predominantly are. And in some of those countries, that makes up a fair percentage of the global population. So you've, off the table, probably removed access to at least the samples, probably the raw data, from these trial participants. And that's something that is going to continue to be a problem for the foreseeable future. I don't see anything changing in that domain right now.
I tend to agree with you. I direct NICHD's Newborn Screening Translational Research Network, where we're seeing a real push towards more federated models where you hold your data within your system, and we find ways of telling you, here's the question, here's the actual data fields we need back. So you control what comes back in. You don't deliver case level data back into a centralized system, but bring back the data needed to answer very specific questions that are de-identified within your data systems. There seems to be a real shift. Global Alliance for Genomic and Health is moving in that same direction with some of its blockchain network systems for transactions and such. So I think more and more tells me we're going to end up in a much more federated world where data lies locally, and we find ways of interacting with local data stewards, really, to get it back in to answer questions.
So just pointing back to that, I would say that there's value, though, that you're missing there with the intrinsic value of the raw data, right?
Oh, I don't disagree.
You've now done some sort of analysis. There's a bias somewhere in there, so I think that's a missed opportunity, but it's probably the best we have right now, right? So
Yeah,
I agree.
I'm into compromise at this point, and there will always be people who will freely donate all their data, and that'll give us a lot of information. But to get over that next hump, we'll have to find ways of getting those more resistant to sharing to be able to play in in an environment where they think their problems have been addressed.
Brave soul.
Yes.
It's okay, it's a general one too. Julie Kerner from Wave Life Science. I'm just wondering how you look at things in the rare orphan disease, where, as we've been talking, you're going to need a lot more sharing and a lot more partnerships there.
I mean, I guess the way I look at it on the diagnostic side, to some of Mike's earlier points, is although the individual disease and diagnoses are eventually rare, the platforms we're used to getting that are incredibly common, right? So I think we're increasingly, many of us, going to a very genomic approach in terms of the diagnostics, and then once we get the diagnosis, everyone splinters off into their own individual rare diagnoses. With that, I think there are, I deal with a lot of N of 1 or N of 5 or 10 disorders, and so it's been remarkable because I think it's only been in this time in history of social media that we've had the ability for patients to really become very empowered and actually start driving the bus in terms of a lot of this. And so before we can even get ourselves organized as either clinicians or as researchers, patients, because they're the ones with the skin in the game, are actually self-organizing themselves. And I applaud them for that. I think that's a wonderful opportunity, and I work with them very much because they find sort of the frontline information, and then I use them, as I act as their translator. I call myself a Sherpa. I sort of guide them along their journey that they're going along and try to learn as they're learning, because I think we're all learning this very much together on the fly. So I think the interesting thing is, though, as we're doing this, I mean, with many of these rare disorders, it absolutely is, they're spread out around the world, except where we have founder mutations in certain cases, but they're spread out around the world. And in a very positive way, we've had families literally come together online to be able to meet each other a few times in person. The hard thing is to be able to, from my point of view, dealing with, for instance, people that are developing treatments, which is ultimately what we want to be able to do, to make sure that folks like you know about everything that we've got on the clinical side, so that as you've got either research programs with certain small molecules or certain pathways, to make sure that you know what's out here, so that you know that we're waiting and ready. And I'd love to think about how we can organize ourselves better.
So I will say that our rare disease unit within Pfizer does a lot of reaching out to communities to try and set up these type of collaborations, because we know, first off, access to these patients so that we can test drugs of interest is difficult. These are small populations. Getting them to enroll in a clinical trial is sometimes challenging. So collaborations, we know, are something we have to do. There's no doubt there.
Well, I'll crassly applaud Pfizer's collaboration with us in supporting the training of clinical geneticists, because that really is very important to getting the kind of data we need about these rare diseases. It has been extremely helpful in growing sort of the workforce that can begin to address these problems. On the last question, I've always had a hard time distinguishing rare and common disease. To me, rare disease has had the stuff with really strong genetic influence. Common disease is these two groups where there's moderately strong genetic influences and another group where the environmental influences are probably even stronger. That moderate category is not small, and it's actually a place where many of there'll be a lot of subdivision of that common disease into subtypes that will be individually rare, and you'll be in the same boat as us. in the rare disease world shortly thereafter. Do you see other aspects of oversight, not just LDTs? As I've looked at things like the 20 cases that justify LDTs, a huge number are issues that really aren't in FDA's purview. Many are educational. While we're going to have a lot of data sharing to do, we're going to have a lot of specialty training across medicine to bring people up to speed about the genomics issues within their areas of specialty. Is that a major problem in the specialists that you're working with in pharma?
So I think we view the whole question differently than I would say any hospital center does. And I came from a hospital group before I joined Pfizer, so I'm aware of their point of view and a pathologist's point of view here. But our concern is really making sure that we can rapidly develop an LDT in response to a new clinical target, and then moving it on, hopefully to an IVD or some sort of appropriate mechanism there if it's something that has to become a companion diagnostic. So that I know is vastly different than what a typical hospital center has in mind when they talk about an LDT.
So I'll answer two of your questions, Mike. So the first question is that, or the second question rather, is that I do think we've got an uphill battle in terms of being able to educate probably two generations of clinicians that didn't really grow up in this information. And so, there are small, I actually look to, and I'll point out that I have some of my students here today. And so I actually think in a good way, it's much easier to teach the younger generation because they've just grown up with this. And I think they're actually the bright hope in terms of being able to also spread the knowledge that they're spreading around what we're doing. And I think we also have to bear a lot of the burden to simplify it enough that we only have a sort of defined amount of information that our subspecialists have to focus on and learn. And I actually think that's an exciting challenge. I do want to point out just one thing that Mike was starting to talk about, because many of you may not be in the sort of down and dirty of the FDA, at least from my interpretation. So the FDA started bringing a lot of these issues up because of 20 cases in which patients had been harmed by molecular diagnostics or LDT specifically, either missing diagnoses or making diagnoses that weren't ultimately confirmed, so false positives or false negatives. And just so that, since we're here in a genomics festival, actually most of those are not at least typical what I think of as many genomic tests. I mean, they may be molecular diagnostic tests, for instance, to diagnose Lyme's disease, but it's not, for instance, that we were sequencing people with Lyme's pathogens and, making those diagnoses. So it's actually, in my opinion, and Mike, you can certainly chime in, really not so much about like the sequencing and sequencing diagnostics, but it's really been much more around very molecularly complex tests in terms of taking, for instance, proteomics or small molecule metabolomics, things like that, and using some sort of black box algorithm to ultimately come up with a risk stratifier, or to be able to say that someone's screen positive or screen negative for ovarian cancer or for something else. And so that's really, for those of you who really focus more on direct sequencing and sequencing interpretation or genome interpretation, I actually, in terms of the use cases that supposedly brought this up, it hasn't been nearly as much in that space as it's been around infectious disease and cancer genomics, at least from the way I see it. So.
Maybe I'll ask a more future-looking question just to get your view. So as I've watched the evolution of genomics and the density of information that we deal with, I can't help but think we look a lot more like imaging and the way it's overseen than most any other area of testing regulation. And if you look at imaging, what you have is an oversight world where FDA has a very strong role in ensuring that those imaging machines operate uniformly, let you see what you need to see. And I think there's a lot of resemblance to what's going on in our knowledge of the genome now. There's a medical specialty around interpretation and ordering of tests in that area, with radiologists and others that order tests and radiologists that interpret them. So I sort of have a feeling that we could move in that direction with genomics, only because the density of the information is so similar to what we see in the imaging world, and much harder to control because we do treat these patients and we do diagnose them, and we don't always know exactly what a variant means, but the key is telling people what you know and what you don't know about anything that you have to communicate to them. So where do you think we're going to end up in the longer term in sort of the oversight of this kind of service?
I mean, obviously, Liz Manfield and the FDA aren't here to be able to comment on this, but I agree with you completely that in terms of the boxes themselves, so the sequencing machines in terms of generating the data, we want to make sure that we're getting reliable data that's coming out of them. I do think it's the practice of medicine in terms of interpreting that information. It's doing that similar to radiologist in terms of the clinical context of that particular patient, knowing things like their age, their symptoms. There are literally thousands of variants that we're seeing coming out of a genome to be able to interpret that, and it has to be within that clinical context, and it's a judgment call. And it's not something that, at least at this point, I mean, we might have fantasies about being able to put it through an algorithm and get the answer spit out to us based on machine learning or other things, but we're not there at that point, and at this point, this is people like, Mike and I and others that sit and wring our hands over which of these variants match the clinical context and explain the clinical situation that we're seeing, and that, to me, is the practice of medicine. I mean, I happen to be a PhD and an MD, but it's using all that information to treat patients as we're doing that. So I don't see any differences actually in terms of radiology.
So I think I will say that if you want to talk about future-looking viewpoint, we would anticipate that eventually you probably are sequencing everybody in a clinical trial, right? At least in the next few years, not just a GWAS or an exome, but probably a whole genome. You may be doing additional tests on top of that, like transcriptome or epigenetics, layering on that data across everything. And at the end of the day, I don't really care how the generated, what box the data was generated on. I just want a quality assurance that generated data is accurate and we're not missing things we shouldn't be missing, and that we're catching everything we should be looking at. So how we get there, I think that's still a very open question, and probably the FDA is still grappling with that, as I think most of us are. So what quality management systems you put in place around that are sufficient to meet that level, and of course clinical care, but without stifling the innovation we've seen in this space. That's the core of our question here, right?
I just want to make the final point to amplify what you said, is that I think it's very critical that we don't stifle innovation. We're not at a steady state yet at this point in terms of being able to interpret the genome, and I think the thing that always concerns me about FDA regulation on this is locking things down, and I just don't think we're at the point that we can lock things down. There's still too much I'm humbled to know that there's still a lot that we're learning in terms of doing this, and we need the brightest minds bringing to bear to be able to solve these problems. So.